So our first speaker today is Dr. Eric Olson, MSU wheat breeder. We are very lucky to have an outstanding wheat breeder at MSU. We were asked, along with other industry partners, to help support the wheat breeder position. It has been great investment for us, for the state's wheat farmers, and to have a bright, young wheat breeder ready to make a difference in the industry. It is my pleasure to introduce Dr. Eric Olson, 10-year MSU wheat breeder, to share with you the varieties he has on the horizon. Dr. Olson.
Hey, great to see you all today. I appreciate the invitation. It's a privilege to speak to you all.
We got to switch microphones.
So at MSU, all of the wheat genetics, the wheat varieties that are available to you all for production on your farms, they all pass through our testing program. Whether that's through the varieties that we develop with our breeding program or through collaborations with private industry, and also our collaborations with our other folks in the public sector. And so we have reciprocal agreements with all the private companies, and where they test some of our material in their locations and we test their material here in Michigan. So all of these varieties are vetted, and we're looking at grain yield, of course, yield potential, and profitability is the #1 factor that we're trying to breed for here. And I want to highlight that varieties they're a part of your management program just as much as your fertilizer or your horsepower and equipment, all of that. They fit in within your program, and they can help increase the profitability by a couple different mechanisms, right? Varieties are different for all of these traits, right? They have differences in disease profiles, resistance packages. Their flowering time is different, right? And I know that all of you are applying a T3 fungicide. We don't forget about Fusarium, right? Years like 2022 and 23, when it was so dry, we didn't see a lot of scab present in the region, but it was across the region. We were a little bit unscathed here in Michigan, but it's important to have differences in flowering date to allow you to get out there to apply, to spread your risk across different flowering dates, because they're all going to get infected differently if they're experiencing different weather. It allows you to get the work done too, spread your fungicide application window, right? And physiological maturity is another trait. That's when the crop is done, when that plant is done photosynthesizing and pumping sugars up into the spike, right, to fill out that grain. And that's going to affect your harvest timing. We have varieties that are a little bit early. Yeah, there might be a slight yield penalty to those varieties, but you don't want to stack all your acres with late maturing varieties either, right? Because that can affect your harvest timing. And as we know, we've had some issues with falling number, right? So a lot of today, I'm going to be talking about the work we've done on falling number, and I want to share with you some strategies to help mitigate that risk. Yes, the risk is there every single year, just like with these diseases that we face, right? But I want to give you some tools with these varieties to help mitigate that risk. And then disease resistance, right? You want to have an insurance policy on some of those acres. A good example of this is the stripe rust epidemic that we saw. It was across the entire eastern soft wheat region, right? We had stripe rust issues, and having that resistance built into your variety can help make the decision for you a little bit easier. Do I spend the money on a T2 fungicide to keep that flag leaf clean? It's an extra cost. Or do I save my fungicide for later, for flowering, right, for that T3? So if you have a clean flag leaf from genetics, that helps to make that decision to save your fungicide application for T3 to help with profitability a little bit, so save you some money, right? So we have a lot of different varieties. This is our breeding program. This is a shot from 2022 where we had, anyway, a lot of differences. You can see all of these varieties are different. Across the board, they're different. And so we don't forget about scab in our program. We don't forget about it. If that risk is there every single year, every Johnson grass and every ditch and every corn and every corn plant in every field can be a reservoir for scab, right? So we inoculate with Fusarium samples from every location that we test. We test across 7 locations, so we farm in 7 counties across the state of Michigan, and we get Fusarium isolates. I say we, it's the folks that do the lab work. I hope you get a chance to talk with Sammy and Amelia and Libby today that make all this work happen. So we cook up our scab inoculum, and this is what's actually happening in the field. We get these little, this black layer here. This is the spores that are going to be firing off of there and infecting the wheat plant. So we deliberately create a disease environment in our nursery. This is on campus here, the plant pathology farm. You all may have seen this if you were at a field day last year. And so what or 2 years ago, I believe it was. So we've got individual rows or individual varieties here, and we're rating for how frequent is infection. We're looking at the incidence, how far that infection spreads within the head, the severity, and then you can use those to create an index. And then of course, the bottom line is DON, right? We want to keep the DON levels low. And the most important factor for managing scab is variety selection. Planting a resistant variety is better than planting a susceptible variety and spending $20 an acre to apply the fungicide. You still need the fungicide with the resistant variety for the best policy for controlling scab. So variety plus fungicide is the best control. And we have a range of scab resistance traits, right? There's a lot, you can see severity, the most resistant lines generally have a low severity, meaning there's very little of that head infected, and a low incidence, meaning that in this case 5% of the heads in that plot were infected. That's pretty good resistance, and generally that translates into lower DON. Right? I hope you never see 20 ppm on your farm, right? That's why we're here, is to prevent that from happening. This is under no fungicides, worst doomsday scenario, okay? And so we see pretty good control in the resistant lines, and we do see it go up to close to 40 with some of the more susceptible varieties. But having a low severity and low incidence doesn't always translate into low DON. It's kind of an antagonism where that plant, the resistance that the plant is putting up is just causing that fungus to produce more mycotoxin, because that's all it can do is produce mycotoxin. That's how it makes a living, right? They're just pushing the fungus a little bit harder. And sometimes you can see visual symptoms, right? They show up with a higher index, but that DON is pretty low. So what that's saying, I've seen this a lot. I don't understand this resistance mechanism completely yet, where you see susceptible heads, but then the grain that is there is completely free of mycotoxin, or largely free of mycotoxin, better than the most resistant lines, right? So we have a different range in resistance, but I'm encouraging you to plant. If you want to go for a susceptible variety, if you know you have a susceptible variety, it's important to have as a partner a resistant variety on your farm. And I know I'm pushing multiple varieties on the farm, and I know you don't all have those logistics. If you do, though, I'm encouraging you to plant those. If you're managing your own seed lot and you're going to be working with your own on-farm seed lot, it would be good to have to purchase some on the side. And if you have been sticking with one variety for a few years, I'm encouraging you to plant a few bags. Just buy a few bags, try a couple strips to see, just try it out to see what there's a lot to choose from, right? So it's a good idea to shake it up a little bit. Now, we got blindsided again this year. Not as bad as 2016 was the last time we saw stripe rust in the state of Michigan. We don't have dedicated germplasm screening facilities like they do in the Pacific Northwest. Stripe rust is an issue every single year for that export market, right? Their soft whites and their hards and so forth. But we have to do a lot of this work in-house. We have had great graduate students working on this in the past. So right at this farm in 2015, Andrew Wiersma collected a stripe rust race that we've been using, and Mel Winchester grabbed this leaf rust race from Mason. So we're working with on both of these. So these are important traits to consider when you're selecting a variety. These are questions to ask your seed dealer. What is the disease resistance profile for these? Do they have stripe rust resistance? So we've gone through 2,400 cones. This was basically my entire so we were inoculating over New Year's Eve and early in January, and so we got the disease. So we have this data to share with you now. And so we've evaluated the commercial yield trial. So all of the commercial lines that are out there, we have recommendations on. And I don't have a comprehensive list for you. We're going to be sharing that with the seed companies here. But what we found is we have the majority. So we looked at, of the 59 commercially available varieties that are on Michigan wheat acres, the majority of those, 34 soft red winners and 7 soft white winners, have disease levels that are low, less than 4, right? So this is a 0 to 9 scale that's going to characterize a level of resistance. So if you have a variety with resistance levels that are on this end of the distribution here, 0 to 3, it's probably going to hold up. It's going to be okay for you in terms of holding off holding off on a fungicide application to the T3, right? Saving your fungicide. Now, if you know something like this, if you've got Dynagrow 9242 out there right now, this is what you're going to be facing if we do see stripe rust again, right? So knowing that, you're going to have that T2 fungicide ready, right? And I know probably one of the most common practices in terms of fungicides is a T1 and then a T3, right? The T2 is less common, right? And so having this kind of resistance can keep it less common, but if you know that you have a variety, even in this 4 to 5 range, that's kind of where varieties like, I'm going to say, like DF271, right? I'm trying to hit some varieties that you all might have on your fields today, locally here. You're still going to need a fungicide on there, right? So these are questions that you need to be asking your seed dealer about those varieties that you have in your field because they can affect the decisions that you have to make, right? Okay, so but we're in a pretty good shape when it comes to stripe rust. I think we're okay overall, right? You just have to be aware of it. Another issue that's present, the risk is present every year, is falling number issues, right? And that's related to visual sprouting. In other parts of the country, it's temperature fluctuations. You may hear of late maturity amylase activity, and that's a fluctuation where you have a drop in daytime high to nighttime low to the next day. When you have a 30-degree drop like that, it causes stress on the crop, right? And so then it responds by metabolizing starch. But what we're mostly working with is related to rainfall events at harvest, right? Right at harvest time. It can be dry, like 2023, right? It was dry all the way up until harvest, and then it rained continuously. It was challenging to get wheat out of the field. So for years since I started, we've been evaluating varieties with visual sprouting, you know, with a low visual sprouting score to a high. And generally that correlates pretty well with what is important to us is falling number, right? It's correlated to falling number. It's an easier trait to evaluate. Falling number is a little bit challenging. We have to handle those samples 5 times from when we harvest them out of the field, to when we miss them, to when we have to thresh them and then grind them and then run the falling number test. It's a worthwhile process for us to be doing that. So to mitigate the risk of falling number, you can use genetics by selecting varieties that maintain a high falling number under these experimental conditions. Right? How we define those really will tell us the result. It determines the result that we're getting. So we've been, I'll talk about that in a little bit. And you can select varieties with different flowering dates and different physiological maturity dates, essentially a different grain fill period. Right? The grain fill period correlates to how early it's flowering and how long that grain fill period duration is going to be. Right? So you can split your acres and have varieties that are ready to harvest while the others are finishing, right? So you can stagger things a little bit. And then here's the message. When you stagger them, that means you can harvest them at different times, and then you can harvest when conditions are optimal, right? And that could mean at a higher moisture percentage. Now, I know a lot of our soft white growers are familiar with this process, and the mills are receiving grain at a higher moisture. It's better to cut that wheat at a higher percent moisture than to let it sit in the field and let that falling number drop. And you're going to be losing 3/4 to a pound in test weight every single day that it gets rained on as well, right? So that's another consideration, right? And you know, I'm afraid the wheat industry has dockages for these. You know, you don't get a premium for a 400 falling number. You get docked for a falling number below 250. So it's important to spread that risk around across different varieties and make sure that you're taking these steps, whatever steps you can, to mitigate that risk, to make sure that we have a high quality crop. Because low quality wheat stays around for a long time. It just drags through the mills and through the process. And DON is easier to blend out than amylase, right? There's an enzyme. When wheat gets rained on at harvest, it kicks off an enzyme, amylase. It breaks down all that starch. And it is harder to get rid of that enzyme than it is to get rid of DON. You can just blend it out 1-to-1 to get to an acceptable level for DON. Falling number is a little harder to deal with. Right? So this has been the process we've used for about 10 years now in evaluating genetics for falling number. We go out to the field, so we walk these plots every day, and when a plot hits physiological maturity, when the peduncle, right, you've got the spike, the peduncle, and then the plant, when that peduncle goes from green to golden, that means their photosynthesis moving sugars up into the spike, it's done. So that plant is done metabolizing, it's done photosynthesizing and filling grain. It's time for dry down. So we take them into the greenhouse and we dry them down for 5 days. So we're sampling everything, whether they're early or late maturity, we're sampling them all at the same time. So they get all the same experimental conditions. So we essentially rain on them in the greenhouse for 3 days. And this is, it's 30 seconds of rainfall every 5 minutes. So they're not getting rained on continuously, but they're kept wet, right? There's dew on those heads. for 3 days, and then we run the falling number on those. We let them dry down, and then we mill them, just a coffee grinder, and then we run the falling number. And then we can push them a few more days to get the visual sprouting, and this is what we would do in the past, just because it's easier. We don't have to thresh and mill everything. But what we're focused on falling number now is the big number. And what we found in the gray circles here, that's our red wheat. And on the y-axis here, 0 to 9, that's a sprouting scale, right? So anything above a 3 here is visually sprouted. You can see that there's sprout damage. And on the y-axis here, we've got our amylase activity. That's a proxy for falling number. So it's just an enzyme test. And you can see the red wheats kind of cap out, right? Whereas the white wheats, they go a little bit higher. And that's because they're producing a bit more of that alpha amylase enzyme. Well, that's our hypothesis. We have some studies to show what is actually happening between the red wheat and the white wheat. But we do know that red wheat will sprout, and the falling number will go down with 3 days of misting. That's what we used this year in 2024 to evaluate all the commercial varieties. We were probably pushing it a little bit far. We only had one soft red that stayed above 250 after 3 days of misting. So we have samples across these different sampling dates here in 2024 to see what is the appropriate date where we can resolve varieties without crashing them all, right? So in 2025, we've got this 8-hour window where we really want to see how much water is getting in there into these. We're going to weigh them, and we're going to see differences. I expect to see differences in terms of how the different varieties are absorbing water in there, because it takes water to kick off all of these enzymatic events, right? To trigger low falling number, it needs water. So we want to see, that's one of the questions we're asking there. And of course, it's Amelia and Samantha and Libby that are doing all this work. And I invite you to discuss this with them. We've got a poster here, and they're happy to talk with you about all this work we're doing as well. So after 3 days of misting, here's the report card for our soft reds. We have some that are in the okay range. We have one that was at 268, 234. If you ran a second test, you probably would get above 250 on that, right? You can roll the dice on that. There's a 30-second variability with falling number 2, right? That's why you can go to another elevator and get a different falling number. It's frustrating working with this test, too. It's not the most precise test. You can get 30-second differences for the same grain sample, right? But what we're seeing is it doesn't necessarily correlate very well with the pre-harvest sprouting score, that 0 to 9 score. We have some that stay below 1, don't sprout, but they get a low falling number. But overall, I think we want to see that average a little higher. So I think next year we're going to be using 2 days of misting. So we're not pushing them as hard as we want to try to replicate what the crop is actually experiencing at harvest in the field. In our white wheats, this was kind of expected. But what I want to highlight here, actually, I want to make sure to highlight this, is here is the difference. I don't think 119 is marketed anymore because it didn't yield as well, because it finishes grain fill a little bit too early. But what I'm showing you here is that you have, in this case, you have a 4-day window, a 4-day difference in when that crop is ready, right? They're going to be when one is at 20% moisture, the other could be at 14% moisture, right? Because of that 4-day difference in maturity, right? We see that in white wheat as well. We see almost a 7-day difference between Jupiter and Moonlight, right? Moonlight doesn't yield very well. I mean, it does, but it yields well for only having 26.5 days of grain fill, right? That's why you see that performance difference between these two types of varieties. But that's just to highlight that there are differences in maturities that you can leverage in managing your crop, in terms of harvest timing as well. Okay, so we know that soft red and soft white have this difference. This is kind of a no-brainer. You're like, yeah, tell me something I don't know, Eric, right? So, but we also saw something very different here. And this could be due to differences in how much water that crop is taking up, because white wheat doesn't have pigment, and the red wheat pigments, they're an oily pigment. They exclude water from the grain. Like in nature, like wild wheats have that red seed coat, and part of the mechanism is it's excluding water, right, from that seed. So we're investigating that. But so specific traits, right? That's grain color is one trait. But on versus onless, we saw the onless varieties might be shedding water a little bit better, right? And this is amongst just within the soft reds, right? This isn't being biased. I don't think there are any oned. There's just that Dynagro that is a soft white that's on. Most are onless. But amongst the soft red, we do see a modest difference between the on versus the onless types, right? So we're with the onless types having a little bit higher falling number. So that's one opportunity to manage, another opportunity to manage falling number with different genetics, different varieties. Actually, I should be pausing to ask any questions or comments along the way. I've been monologuing for a few minutes here. Does anybody have any comments or questions on this? I'm sure you've got some. Yes?
I just want to compare England to the US. So for your milling, what is your spec for the falling number?
We're looking at a minimum of 250 is going to be the spec. And
Repeat the question.
yeah, the question, he's asking what is the spec for falling number in the US and relative to the UK. And so we're looking at about a 250, right? And there could be different specs depending on the flour product, right? The mills might have different specs depending on their customers, but the value to be delivering is 250, right? And we can see a baseline with no rainfall. You're going to see a range from 275 up to 400, or maybe even 450 for soft wheat in the absence of any rainfall, right? So there's a lot of variation just in baseline falling number, right? But it's the rainfall for us that's largely metabolizes all those large intact starch molecules and affects the quality then. That's a good question. Any other comments or questions? Yes, sir.
If they're testing for this at the elevator,
Yes.
are they doing a good job, a fair job, or a poor job?
Well, that is entirely subjective, right? The question is, are they doing? It's a loaded question. Are they doing a good job, a poor job, or a fair job at the elevator? And I'm just going to say they're doing the best they can, right? Because we're working with the same technology, the same falling number. It's like you're not going to get rid of test weight as an international metric for grain quality, just like we're not going to get away from falling number. It's been there since Sven Hagberg invented it in the late 1960s, and it's embedded in the grain trade right now, right? We're working on other tests that might be more accurate, and in fact, Washington State University has gotten, through ARS and through some federal funding, have developed a rapid test, right, for falling number, and they've coupled it with DON, so you can get DON and an amylase test on the same dipstick, right? It's the same kind of it's a color change test, right? Same concept. We're in the same kit. The problem is that they just have green, red, and then yellow, right? It's that yellow, right? That's where right around 250. It's between 275 and 225 where you get that yellow range, where, and that's the hardest place to work. So, yes, another question. Up to 450, that's right.
Is that variety dependent or year dependent or?
Both. It's both, right? So you can take a hard wheat is going to have a higher falling number, higher baseline falling number anyway, just because of all of the gluten proteins, right? Glutenins, gliadins, and all. It's just because of the more protein, right? And we've tried to hack the falling number with different quality traits, right? Can we have just like a high lactic acid SRC, which means really strong protein, strong gluten for a crispy cracker? It doesn't seem to hold up, right? But you can have in hot, dry years where you're going to have a little bit more protein, where overall the crop goes to from, maybe 9 to 10.5, maybe 11%. Those are going to have a higher falling number as well, right? So that's environment and so a lot of it's environment, but you can have some genotypic variation. But we don't see any genetic variation just at baseline falling number. So in the environment, I think would play a bigger effect. Very good, very good. Does that answer it?
Well, what I was getting at is there some variety, I grow the white wheat.
Yes.
Is there a variety that maybe gives me a little bit more higher starting point that, if I start at 275, I only got 25
right.
wiggle room. If I can start at a higher 400, then maybe I got more wiggle room.
Right, right. They exist. So Jupiter and Whitetail both have the highest starting falling number, but they also have, after 2 days, they also fall the hardest too, right? So even though they might start high, they fall pretty fast. And I've got, but we do have soft white winter wheats in our breeding program. We have been using genomic predictions. We've been phenotyping pre-harvest sprouting for almost a decade now. We have so much data that we're using to train genomic prediction models. Anybody in the animal world who works with Zoetis for heifer replacement, herd improvement, right? You can predict feed efficiency and milk yield, we can predict falling number, right, and pre-harvest sprouting resistance. So we do have some varieties that will tolerate 2 days of misting, right? Once you hit 3 days, they all kind of fall off. I'll show you some figures on that, actually. So we did something called an on-off-on experiment to try to replicate rainfall followed by drying and subsequent rainfall, right? What we've been seeing in production. It's not perfectly going to match what's in production, but it's a close experimental approximation. And so 1_1 means it's misted for 1 day. We pull it out of the dew chamber, pull it out of misting for a day, and then mist it for another day. And most of the soft red, so these were 5 experimental soft red winter wheats from within our breeding program, because we had enough material that we could sample a lot of spikes. Because we need 7 grams of flour minimum for every falling number run, so we had to get a lot of grain. But you can see that it tolerates up to 4 days, right? We end up to, so 212 would be 4 days. And the majority, there's a big distribution, but a lot of those varieties, there's 5 different varieties with 3 replicates nested in here. Some of them are staying above 250, right? So some of them are able to tolerate that. It's a different scenario. The soft white wheats that we evaluate, that's a bit of a log curve there, right? Only with 1 day on, 1 day off, and 1 day on again. We see this huge range, and they all collapse after a few days, 2 days of misting. Most of the soft whites they fall apart. But that's a pretty big range, right? Some of these varieties, this is my comment about varieties. So these are 5 soft white wheats that were variety candidates last year. So a few of them have fallen out. But what we see is some that really tank, right? This is just after 2 days, 1 on, 1 off, 1 on again. Some are intermediate, right, and mixed, a little too close to 250. But we have two different varieties, 212. Both of these lines, and 193, both of these lines were cut from the program for various reasons. But 212 has got a SIB line, 213, that's still in commercial yield testing this year. And they perform nearly identically. One just has a little bit better scab resistance. So we do see results from our breeding efforts for pre-harvest sprouting resistance. So 10 years of efforts, we do have varieties, soft white wheat varieties that can have a higher falling number that resolve from those with lower falling numbers. So I feel like we've been making progress. So they are out there, and that's the rationale for continuing to screen all of these varieties. So that's just an overview. We have 8 years we can get up to 4,000 acres of certified seed, if that was needed, right? So we've got a very fast breeding program. And from the 2022 cohort, this was in 2019, crosses we made in 2019, after 3 years of replicated testing. And we use 25R40. It's probably the most widely planted variety in the yen, right? That's why we use it. Cy 100, I don't know if it's the US company that markets Syngenta products has a different name, so I don't know if they're actually marketing it, but it's all over the place for our checks. So we have good yield comparisons to there. So here's that SIB line that has the 212 that had really good falling number, sprout resistance. It has a good foliar package. We've got leaf rust here and stripe rust, but it's pretty susceptible to scab. But the yield is there, and I might end up culling it just because of that test weight. That's kind of a basement for test weight. Anybody that's grown Cy 100 is familiar with that test weight issue. Now, we've got a couple, in asterisk here, these 2 lines have the FHB1 gene, right? So you can get a pretty high level of resistance in soft wheats, right? Soft red and whites with this gene. And it's a good question to ask your seed salesman. Does this line have the FHB1 gene? Because if you know you have that, and you're putting down a fungicide, that's the best policy for mitigating scab risk. And it's a major target for our breeding program as well, and every soft wheat breeding program in the country. Everybody's very focused on scab, right? So we have R97, that's got an upright canopy, very, very good for a narrow row system, right? We've got a good foliar package, so that's going to be a good organic variety as well. R57's just got really high yield. It's intermediate for scab. It's just been performing at the top of the trial. That's why it's still there. So we've got some good lines coming up out of the program. I want to thank everybody for the support. This is great. We couldn't do this without support from you all, from Michigan wheat farmers. They're making it all possible. And this is Amelia and Libby. There's Amanda who manages our program here, and John's a grad student who's working on a lot of cool variety development activities. Just want to say thank everybody for the support. And I appreciate y'all being here. It's a privilege. Any other questions or comments?
Time for 2 questions.
Yes.
number actually relevant to the flow industry?
It is. It is. It's quite relevant because it affects the marketability of that flour, right? And 95% of all the bushels produced in Michigan are milled into flour regionally. If not here, it's in Toledo. I don't know how much goes to Kentucky, but it's going to be most of that. All of that is relevant to the milling industry in Michigan, and so they have customers that have very strict flour specs, like a company that's going to, you know, if they're going to make Oreos or you know, any kind of, or saltines, right? There's a very specific flour spec that flour has to meet. The grain that they're buying to mill has to match those specs. Sometimes there's flexibility, right? And I had witnessed a very great discussion about the impacts of test weight on actual. There was no impact of test weight on milling and baking quality, right? And so there was a bit of a discussion about that. But what I'm saying is it's important because it affects the marketability of the crop and just the flow of that crop through the entire value chain, all the way from your acres, from your farm, all the way to the supermarket. You know, it affects that flow of grain across the value chain, I think is the best answer that I can give on that. It's a very, very, very relevant question. Good, yes?
So at what moisture do you think you need to start worrying about if it rains, we're going to have falling numbers?
Okay, okay.
Also, like, is it way worse at 15 moisture than it would be at 20? Does it keep getting worse?
Right, so if you get, it depends on physiological maturity, right? And so at physiological maturity, that grain is going to be 23 to 24. So it's going to be a little bit higher, right? And so when the risks really starts when that crop, when that peduncle goes from green to golden, right? From green to yellow is when that risk starts. So it's not as much the grain moisture percentage, it's just that developmental stage for the crop. So that's something when you're scouting, right? If you've got acres spread around and you're scouting, that's what to look for. That's what to look for. But it is important to check the moisture as well too. Now, we don't have that kind of data yet to say that if your crop was at, let's say, 14% and it got rained on, is that different from if it was at 18%, right? Those are interesting questions, and what that's going to affect is how much water that grain is going to take up, right? That 14% crop is going to pull in more water than just by osmosis, right? It's going to pull that water in more so than an 18% crop. So it's a relevant question and a good question, but we don't have data to answer that just yet.
You're more so looking at the greenness of the actual straw.
Yes. When the straw just below the spike goes from green to yellow. And it's hard if you're not out there walking plots every day. You may not walk it every day, but it's something to keep an eye on then. Yeah, and that's when you can start tracking your moisture, right? And depending on how many fungicides you use, your canopy could be a little bit different than what the actual spikes are too, right? So I'm sure everyone has noticed that difference where you can have a green canopy and your spikes kind of go golden. So you might be at physiological maturity before that crop canopy is actually straw colored as well. So yeah, good, important considerations.
Okay,
All right.
thank you very much, Eric.
Thank you. Thank you, everyone.